So as you probably already know, microplastics are defined as from a variety of different sources. These can include larger bits of plastic or macroplastics, as they're known, such as plastic bottles or carrier bags that are broken down into smaller and smaller pieces by UV degradation and wave action once they're in the sea or on beaches. There's also pre-production pellets, or nurdles as they're called in the UK. I don't know if that's what they're called anywhere else in the world, but we call them nurdles. And they're plastic in its raw form before it gets turned into anything useful. So these lentil-sized kind of pellets are transported around the world in shipping containers and can enter the sea as a result of cargo spills or industrial mishandling. There's microbeads from cosmetics such as shower gels and toothpastes, and these are tiny flakes or spheres of plastic that are too small to be filtered by water treatment works, and they can enter the sea from household water, wastewater. Also microfibers, these can be released from synthetic garments during washing and can also enter the sea in a similar way to the microbeads through the wastewater. So one study found that for an average 6 kilo load of washing, over 700,000 fibers could be released per wash. And consequently, they are the most common type of microplastic that we find in environmental samples. There are also lesser known sources of microplastics, such as fragments of vehicle tires and road marking paints, which are actually made of plastic. And they kind of, as the tires and the road paint kind of wears down over time, bits break off, and they can enter the kind of drainage system, which is not filtered at all in most places. And obviously through rain, water and runoff, it can enter the environment that way. But they're kind of more kind of emerging sources of microplastics that are being investigated. So once in the marine environment, microplastics are known to be ingested by animals at the base of the food chain. So this top image shows a copepod, which is a type of zooplankton, which has consumed fluorescently labeled microbeads, which are these green patches here, which were ingested during a lab feeding experiment. And this bottom picture here shows a wild thick-backed sole larvae, so a fish larvae, which has ingested a microfiber. So inside this red circle here is a tiny microfiber, which was found in the digestive tract of this fish larvae. And the fish larvae was probably only a couple of days old when it was caught and sampled. So research has shown that microplastic ingestion in these low trophic level organisms can lead to a reduction in feeding capacity, energy reserves, reproductive output, as well as detrimental alterations to intestinal function. But it's not just the physical presence of microplastics that cause these problems. Chemical contaminants present in the seawater, such as PCBs, have the tendency to absorb to their surface. And these and other chemicals added during production, like plasticizers, can desorb into the biological tissue if ingested by an organism. And that can cause detrimental effects on their health. So due to the small size and omnipresence within the world's oceans, microplastics are available to be ingested by a whole range of animals, from the microscopic zooplankton that we just saw to larger animals like fish, turtles, seabirds, and marine mammals. And it's these guys that I was interested in for my PhD. So why was that? Well, firstly, they're diverse. As you can see from this picture, marine mammals come in all shapes and sizes and exhibit many different ways of foraging, communicating, living, and socializing. And this variation means that they can respond differently to human activities. Many of them are also long-lived, which allows us to observe how they adapt or don't adapt to changes in their environments. Unfortunately, this can also mean that they're vulnerable to population declines due to their low reproductive rates, and many are of conservation concern. So, for example, this bowhead whale, well, not this one in particular, but bowhead whales are known to live up for over 200 years. And so also many marine mammals are also top predators, which means that studying them can help us understand whether or not the ecosystem that they live in is healthy. And this is particularly important for the marine environment because it's so challenging for us to monitor. And last but not least, personally, I think marine mammals are super cool. So why wouldn't we want to learn more about them and protect them if we can? So there are two main ways that microplastics might be ingested by marine mammals: direct or indirect ingestion. So direct ingestion can occur as a result of accidentally consuming microplastics that are in seawater. This may happen during prey capture. And although this image shows a picture of a dolphin, this pathway is more likely to occur in less selective feeders like baleen whales, who engulf large volumes of water. So it would also be a mechanism by which other kind of filter feeders might ingest microplastics. So for example, like whale sharks and other large filter feeders. So the indirect route of microplastic ingestion is via trophic transfer. And this occurs when microplastics in seawater are eaten by a fish or other secondary consumers. And then they are in turn consumed by predators such as seals and whales and dolphins. And the microplastics are then passed up the food web. And although this seems like a fairly obvious process, it hadn't really ever been observed in top predators, only kind of low trophic level organisms like crabs. And during my PhD, I sought to examine this. So with the help of some colleagues that worked at a seal sanctuary near to where the university is here in Cornwall, I analysed the scats of captive grey seals and the wild caught fish that they were fed upon. And both of those had microplastics within them. And this is important because the seals are captive. They're not exposed to plastic pollution like wild seals are. And so the presence of the microplastics in their scat or their feces indicates that the trophic transfer of the microplastics from the fish had occurred. So if the fish contained microplastics and so did the scat, and there's not really any other way that the seals could be exposed to plastic. So it kind of had to come from the fish, really. The next step of my PhD was to examine the extent of microplastic ingestion in wild marine mammals. And I wanted to ask questions like, how many microplastics does an animal consume typically? Once ingested, do the microplastics get stuck anywhere in the digestive tract, or do they pass through? And if they do get stuck, do they accumulate? And if so, where? What type of microplastics are exposed to? Are they fragments or fibers? What do they look like? How big are they? What color? What are they made of? What polymer type are they? Knowing this kind of information can help us understand where the microplastics might be coming from. So to answer these questions, I examined the digestive tracts of 50 animals from 10 marine mammal species that are stranded around the British coastline. These were an Atlantic white-sided dolphin, bottlenose dolphin, common dolphin, grey seal, harbour porpoise, harbour seal, pygmy sperm whale, Risso's dolphin, striped dolphin, and a white-beaked dolphin. And the coloured dots on the map correspond to the species and where they stranded. And as you can see, the common dolphin and harbour porpoise were by far the most numerous. We had 16 common dolphins and 21 harbour porpoise, and this kind of reflects the abundance of those species in British waters. They're kind of some of the most common that we have. This is a bit of background information. So the Cornish animals, so all of this kind of work was very collaborative and involved many different people. So the animals that stranded in Cornwall, right down here in the southwest of England, were supplied by the Marine Strandings Network, which are part of a local conservation charity, and the post-mortems of the animals carried out by qualified vet James Barnett. The Scottish animals came from the Scottish Marine Animal Stranding Scheme. Again, qualified vets did the post-mortems, and then the rest of the animals came from the Cetacean Investigation Programme at the Zoological Society of London, and the post-mortems were carried out by Bill. So when an animal stranded, a post-mortem was carried out and the digestive tract was removed. The digestive tract was given to me, and then I extracted the content of the stomachs and the intestines in a highly controlled environment. So this kind of area that I'm working in is called a positive pressure laminar flow hood. This limits any kind of airborne contamination. One of the things we face as microplastics researchers is that there are microplastics everywhere in the air that's surrounding us right now. You know, all of our clothes, the things, our seats, the carpets, everything is made from plastic, and all of those things can contaminate our samples. So we have to work in this kind of this flow hood. Obviously, I'm wearing plastic, but that was for health and safety reasons. There's lots of zoonotic diseases that can be passed from marine mammals to humans, so I had to wear some personal protective equipment. But we took samples of this plastic to compare with our results to make sure that anything found in our samples didn't originate from the plastic used in the lab. We tried to limit the amount of plastic used in our equipment. So most of as you can see here, my tray is made of metal. I've got a beaker made of glass, and all the implements were metal. But obviously you do have to use some plastic, but you can control for that in your kind of result. So once I'd extracted the gut content, I used an enzymatic digestion to remove some of the organic material, or as much organic material as I could. I then filtered the remaining fluid across a mesh disc using a vacuum pump and inspected it under a microscope. So you can see here, this is the kind of mesh that I ended up with. You can't really see anything on it, but once you sort of look down a microscope, you can kind of pick out anything that's kind of oddly shaped or brightly colored that might be synthetic. And then to verify whether those potential microplastics were actually made of plastic, we use a process called Fourier transform infrared spectroscopy, or FTIR, to determine whether or not the particles are plastic and give us information on the polymer type. So here are a couple of examples of the microplastics I found in animals. As you can see, they come in a range of sizes, colors, and types. They're made of 4 different polymers. We've got nylon, polyethylene, PET, and phenoxy resin. And I'll go into more detail about the polymer type in just a minute. This graph shows that the number of microplastics along the bottom and the frequency on the side. So in total, I found 273 microplastics in the 50 animals that I looked at, and every single animal contained at least 1 microplastic, with an average of 5.5 microplastics per animal and a maximum of 12, so not massive amounts. I only found macroplastics in 1 animal, and this was some green netting in the stomach of a common dolphin. But other than that, I didn't really find anything bigger. So this graph shows the gut section along the bottom, or the gastrointestinal tract section, the stomach or the intestines, and the number of microplastics at the side. And as you can see, we found more particles in the stomachs than in the intestines, indicating that the stomachs may act as a potential retention site of the microplastics, but the relatively low number overall demonstrates that the particles may be able to pass through the digestive system and don't really accumulate. On the other hand, we don't know what kind of sublethal effects the microplastics or the chemicals contained on or in them are having on the animal's health. So this graph shows the cause of death category along the bottom. Because they were post-mortemed by qualified vets, we were able to get an indication of what actually killed the animals. So we've got infectious disease, trauma, or other. And trauma can be anything from kind of boat strike, bycatch, bottlenose dolphin attack. But the animal's health generally wasn't compromised if they were classified as being killed by trauma. And we found that animals that died of infectious diseases contained more microplastics than those that died from physical trauma or other causes. We don't know the biological significance of this finding yet, and further research is definitely needed to understand the impacts of microplastic ingestion on animal health. It could just be that the animals which are already compromised due to ill health feed abnormally and then are more likely to ingest plastic. But again, we need to treat this kind of result with a bit of caution, really. It's quite complicated to unpick. So this graph shows the size categories for microplastics along the bottom in micrometers, and the number of microplastics in each size class at the side. The purple bars are fibers and the grey bars are fragments. Most of the microplastics I found were fibers with an average size of 2 millimeters. And this correlates with other studies looking at microplastics in the general marine environment and other organisms like fish. In terms of colour, black and blue were the most common at 42.5% and 26.4%. And again, this correlates with results from other studies looking at the wider marine environment. Blue and black just tend to be, and clear do tend to be the kind of most common colours we find, probably because those colours are very popular in the use of textiles and other kind of products that are made of plastic. Nylon was the most common polymer type at 61.2%, followed by PET and polyester, both at 10.2%. And many everyday objects are made from these polymers. So nylon, for example, and polyester are frequently used in our clothes and other textiles, ropes and fishing line, even toothbrush bristles. And PET is widely used for plastic bottles and food packaging. So after my study on microplastics in the digestive tracts of marine mammals, we started to think about the role that diet plays in how many microplastics these animals are exposed to. And if I can bring your attention back to the earlier slide about trophic transfer, it looks as though this pathway could be a major route of microplastic ingestion from marine top predators, but we don't know how the type of prey consumed affects this level of exposure. So to investigate this, we created a method which combines DNA metabarcoding to determine the prey composition from seal scats, i.e. what they've been eating, with a microplastics extraction technique. And in this case, we used grey seal scats from a colony in Scotland. At the moment, we've only developed the method, and the next step would be to apply this technique to systematic sampling regime to see, to investigate whether this aspect of trophic transfer could be looked at more closely. And although we developed this method for seal scats, it could be applied to other predators where the question of microplastic ingestion is of interest, even terrestrial environments where microplastics are now being detected. But yeah, it does, from our initial study, we only used 15 scats to develop this method, but from our initial results, it looks as though there's certain types of prey may be linked to the levels of microplastics that the animals are have in their scats, and therefore that they're exposed to in the environment. As with any research projects, our study on microplastics in marine mammals raised more questions than it answered. So here's just a rundown of things future research needs to look at. So the big question that everyone wants to know is, what are the impacts on animal health from microplastic ingestion? And at the moment, we just don't know, but there are three main aspects to consider. So the first is probably the most obvious, mechanical. So what kind of effect on is the physical presence of these plastic particles having? Considering that large marine vertebrates like seals and dolphins regularly consume indigestible items like fish bones and squid beaks, you'd imagine that invisible particles, tiny bits of plastic, wouldn't have that much of an impact. But we don't really know whether these very, very small plastics, known as nanoplastics, are able to pass through the gut wall and into the bloodstream, make their way to organs such as the liver and the lymphatic system, which is responsible for immune function. So, and additionally, an experiment in mice found that nanoplastics affected the gut microbiome and caused damage to the intestinal barrier function. But the trouble is, at the moment, nanoplastics are really hard to detect, and we don't have the technology to investigate this in wild animals yet. But it's definitely a route that needs more investigation. As I mentioned earlier, chemicals that are on or within plastics can be released when they're ingested into the animal's tissues, but we don't really know whether microplastics ingestion exposes animals to these chemicals, or how these microplastic ingestion exposes these animals to chemicals aside from their additional seawater and diet inputs. The relationship is really difficult to disentangle because there's more to it than just X number of microplastics equals Y amount of toxins. There are many different variables which may affect this relationship, such as type of polymer and the age of plastic, but also there's a lag between chemicals accumulating in biological tissue from the plastics. In the meantime, the plastics may have already passed through the digestive system and been excreted. So even if you looked at an animal, it's only a snapshot, but those two snapshots of chemicals and plastics don't necessarily match up. So also plastic in the environment can be colonized by a diversity of bacteria, and consumption of plastic could lead to an increased exposure to pathogens. We don't know yet how this could affect animal health, but a study on coral reefs found that likelihood of disease increased from 4 to 89% when corals were in contact with plastic pollution. So as you can see, we're only just scratching the surface of what plastic pollution means for marine mammals, and there's a long way to go before we have the answers. So in summary, though trophic transfer is an indirect route, it may represent a major route of microplastic ingestion from marine top predators. And when we examined wild animals, we found that microplastics in every one, but the overall abundance is low, suggesting that they can pass through the digestive tract. However, higher numbers were found in the stomachs, and that may indicate that the microplastics can get stuck for an unknown amount of time. From our molecular work, it looks like the extent to which marine mammals are exposed to microplastics is linked to diet, but much more work is needed to examine whether microplastics cause any detrimental impacts on animal health. As I mentioned, this work has been a collaborative project, and I'd like to say a huge thank you to everyone involved in these studies. It would not have been possible without their input, and it was great working with all these amazing people from so many different organisations. So for my PhD, I was based at Plymouth Marine Lab, but also collaborating with the University of Exeter, and then supported by all these other organisations. So thank you very much for listening, and I'll take any comments or questions you might have.